Assalamu alaikum wa rahmatullahi wa barakatuh, dear students. I hope you're all doing very well. Today's lecture is about gram-negative bacteria, and I want you to pay special attention to this lecture because major groups of bacteria of medical importance are actually from this group, the gram-negative ones. And because there are so many gram-negative bacteria that are very important and that you're going to also face when you work in the lab or when you become future doctors, this becomes very important because you will notice that what you learn in this lecture is just touching on the gram-negative. It's not actually nothing near perfect or nothing that is coming to make it more comprehensive of the true number of bacteria in terms also of the clinical importance and so on. So, inshallah, in the next few years, when you learn through the systems, you are going to learn more details about these bacteria. And of course, the clinical experience that you're going to acquire throughout your training, you're going to learn a lot more about them. So in this lecture, we are going to go through these objectives. Please note that there is one less objective than the one in your manual because I have modified this lecture during the making of the slides. So please pay attention to these four important objectives. Now let us start. So in this lecture, we are going to go through gram-negative bacteria. And as you by now know very well that the difference between gram-positive and gram-negative is basically the difference in the staining. So gram-negative is actually means simply is that the bacteria has a different type of cell wall or the different structure of the cell wall that makes it does not retain the purple dye, and so it's going to be washed easily when you apply the alcohol, and eventually it's going to take only the counter stain, which is the saffron stain, so it will give you pink color or red color. But you have to also understand that gram-negative bacteria are the major groups. So what are the two major groups? Is the gram-negative cocci, gram-negative bacilli. But also there is a third type, which is the curved or the twisted bacteria. In this lecture, the main focus is going to be about the bacilli, but I'm going to touch on the gram-negative cocci very briefly. That does not mean that these bacteria are not very important. Actually, they are very important, but they are less common. So the two major groups in the gram-negative cocci is Neisseria species, and the main two Neisseria species that we are going to discuss here is the Neisseria meningitidis, and from the name you can conclude the most important disease that this bacteria cause, which is meningitis, which means inflammation or infection of the meninges. And the meninges basically means the very thin layer that surrounds the nervous system, mainly the brain and the spinal cord. And the second species of the Neisseria group is gonorrhea. Neisseria gonorrhoeae, from the name itself, you can predict that this is a bacteria that can cause a disease called gonorrhea. So gonorrhea is the term used to describe a sexually transmitted disease. And this disease is actually produce different types of symptoms in females than in males, but we're not going to touch in these details now. We are going to learn about this bacteria when you come into the systems in the next semester and next year as well. And when I describe the different structure or the biochemical tests that we use, I'm going to collectively talk about these two together. So whenever I'm talking, I'm not going to mention them separately unless I indicate so. So what is Neisseria species? You can see from the staining, this is actually a picture of a gram stain. And the most important finding in this picture is that you can see gram-negative cocci, and these are actually inside the cells or intracellular. So what you see here in the pictures are very small cells that are found inside polymorphonuclear cells that are part of our immune cells. And actually what you see here is intracellular cells, and these bacteria cells are diplococci, which means that they are present in pairs, so two together. This is what diplococcus or diplococci means. So diplococcus, one pair, diplococci is the plural for this bacteria. So this is the gram stain finding. And when we grow it in culture media, we actually select for a very specific type of media that actually required for this growth is actually this enriched media, which we call chocolate agar. And I'm sure that you have learned about this agar from before. So chocolate agar is basically similar to blood agar. It's nothing to do with chocolate. Actually, there's no actual chocolate in it. Just the color of it seems like chocolate, but it's actually not chocolate. It's basically heated blood. So blood, when it's heated, it lyses or breaks, and then it releases the heme, and then it gives this brown color. So the idea of heating the blood is actually to make the nutrients released from the RBCs or red blood cells, which we use for these agars, actually animal blood, not human blood, of course. We use horse blood or sheep blood. So this bacteria here is Neisseria, requires this chocolate agar to grow, and it has to be also incubated under very specific circumstances in which we need oxygen, but we also need special requirements of carbon dioxide, which is 5% in the atmosphere, and this is something that we can adjust in the laboratory using specific special incubators. That's why we call this bacteria fastidious, because it does not simply grow in any simple media. Actually, it needs special requirements. That's why we call them fastidious. This, of course, applies for both species, Neisseria meningitidis and Neisseria gonorrhea. Now, we come to the biochemical characteristics of Neisseria species. You have to bear in mind that this bacteria is actually very serious, especially the Neisseria meningitidis ones. And when the patient comes in the emergency room, we actually need very speedy diagnosis for meningitis, because it's very severe condition and that you need to treat within minutes, actually, or hours, or otherwise the patient might deteriorate or die. So one of the basic tests that you can do actually on the bench in our lab is called latex agglutination test. So basically it's a test that can detect the capsule that is found in this bacteria, and it's a polysaccharide antigen that is found on the surface of the bacterium. So what you use is a spinal fluid, or we call it cerebrospinal fluid. It's a special type of specimen that we collect from the nervous system. And I'm not going to go through the technical details on how to collect CSF for short, but actually we are going to focus on the microbiological and biochemical part. So we take specific amount of cerebrospinal fluid, and then just a drop. You add this antibodies, and then the antigen is going to be detected in the spinal fluid. And what happens is that it's very rapid result. It will give you agglutination. So agglutination, as you can see from this picture, basically means when you add the antibody to the antigen, there will be a reaction, and this reaction will cause agglutination, which is similar to clumping reaction. And you can see in the picture the difference between a positive and a negative. So in the negative, you have very smooth, homogeneous liquid, but when the test is positive, it's actually causing a reaction that is shown in the clumping. Another very important test we use is oxidase test, and it's actually a test that we use for Neisseria, and it's oxidase positive. Oxidase, as you can see here, is actually a test that detects the enzyme cytochrome C oxidase. So any bacteria that has this enzyme, it is going to be exposed to a compound. We call it phenyl inidiamine that turns into purple or black, and that's as a result of the oxidation process for that is happening in the reaction, and it will give you a purple color. So these are the most important characteristics or takeaways from the gram-negative cocci. Now let's move to the rest of the lecture to the gram-negative bacilli. So in gram-negative rods or bacilli, we are going to focus only on medically important species. What this means basically is that there are so many large groups that are found in the environment or in the animals or plants that we're not going to discuss. We're going to discuss the relevant to the human beings. The first group is called Enterobacteriaceae. So Enterobacteriaceae is the family. It's like a tribe, big family. And I use these mnemonics, which means the abbreviation, to remember the major groups, which is yes picks. So what this yes picks is actually an abbreviation of the main species within the group, within this very big family of Enterobacteriaceae. So we have Yersinia, E. coli, Serratia for yes, and then we have Proteus, Enterobacter, Citrobacter, Klebsiella, Salmonella, and Shigella. And as you will notice in this lecture that we are not going to go through all of them. Actually, it's just a selection for the most important ones that are commonly found and seen on a daily basis in our laboratories and also in the hospital. The second group is the non-Enterobacteriaceae. So it's just non-Enterobacteriaceae. We don't have any other special name. And this contains Pseudomonas species, Ortodella pertussis, and Haemophilus species. I know today you're going to learn so many new names. Don't worry about the spelling. You're going to learn them as we go through. And then there's the third type, which is the curved rods. So it's also bacilli, but they are curved or twisted. And this includes mainly Vibrio species, Campylobacter species, and Helicobacter species. So now let us move on into more details about the first one, which is the Enterobacteriaceae in the coming slide. Well, now we should be going through more details, which is the general features of Enterobacteriaceae, which means that all the group of bacteria in the yes-pix groups actually share common characteristics. First one is that all of them are gram-negative. And the second one is that actually all of them are commensals or part of the large and small intestines. And that's where the term intero comes from. It's actually the Latin term where it means the intestines. And then you are also going to see that these are motile, which means that they can move, except Shigella and Klebsiella. These are non-motile bacteria. And most of them actually are non-fastidious, and the ones that are fastidious we're going to indicate, otherwise it's all non-fastidious. They are aerobes or facultative anaerobes, which basically means they require oxygen, but in the absence of oxygen they can still survive. But actually the difference between aerobic and facultative anaerobic is that these bacteria usually prefer oxygen. If they don't find oxygen, that's fine, but the growth is optimal and better in the presence of oxygen. The other characteristics that all of them ferment glucose, and throughout this presentation today you are going to learn about the different fermentation processes, because it's not just glucose. Each bacteria sometimes prefer also some groups of other sugars like lactose and so on. We are going to learn more about them. And also they produce acid when this fermentation happens. And as a result, this can be detected in the growth media that we use, especially if we are using special agar or broth, liquid broth, there will be change in the color, and that can be detected easily. Another important, very, very important test that can actually give Enterobacteriaceae their special feature is the oxidase test. All of them are oxidase negative, which is again, it does not turn the color of the reagent into purple. It actually remains neutral, and it's all catalase positive. Now, catalase is the test that actually converts into, the hydrogen peroxide is converted into water and oxygen, and then it produces bubbles in the reaction. You can do it on the slide or on the tube, but the reaction will be similar, and you can detect it will be positive. You see the bubbles. The other characteristic is that all of them are non-encapsulated except Klebsiella. So it means that all of them do not have a capsule, but only Klebsiella, which is Enterobacteriaceae, but it has a capsule. So these are very important characteristics that will apply to all. Now, in this slide, I want to teach you about a very important test that actually tells us a lot about the different biochemical reactions or metabolic pathways that these Enterobacteriaceae use, and how we use this characteristic to diagnose and also differentiate between the different groups. One of the most important tests is some test we call Hugh-Levson test, which is a reaction or a test that detects two important reactions. One is the oxidation or oxidative fermentation, and the other one is fermentation. So we call it oxidative fermentation. So the media is designed in such a way that can show us both whether oxidation happens or fermentation. So the test is for short called OF test. So it will tell us whether the bacteria has anaerobic or aerobic ability to break down the glucose. Because for the aerobic, when oxygen is utilized, it's a different pathway than when the anaerobic, where the bacteria actually uses the sugar to ferment it to produce energy. So that's the basic difference. And to make it easier, look at this picture here. We have the media, which is actually basically, you can see it green color here on the left hand side of the picture under the control. We have two tubes. You can see that for each reaction we have two tubes. Why? Because one tube is actually empty. Sorry, it has only the media, and it's on the top, it's exposed to air, so the tube is actually open. It does not have a cover or a cap. The other tube actually is having on top of the media we have a layer of oil, and the purpose of adding oil on top is actually to prevent the bacteria from getting exposed to air. So now we are creating the anaerobic condition. So if you remember when I spoke about uh, the pickle, similar at home, that we add a layer of oil on top so that we prevent air from reaching into the food, and then it will prevent the fermentation, I mean the spoiling of the food. Here it's similar. We want it to be anaerobic. So what is the difference in the interpretation of the test is that when you apply the bacteria inside the tube, or you stab the tube with the bacteria, if it turns into yellow, then it tells us that this is an Enterobacteriaceae because it actually has the ability to ferment glucose. So the media will turn into yellow. And then why it turned into yellow? Because actually what we have here is that we have a specific dye that we add with a pH indicator. So the pH indicator will detect the change in the pH, and then it will change the color. So all Enterobacteriaceae, not just E. coli, they are aerobic or facultative anaerobic. And so because of that, they can also oxidize whatever the oxygen is available. So that tells us why E. coli is able to grow with the tube without oil. But again, it can grow if there's no air, which means the other tube with the top layer of oil. So this also tells us that it's aerobic and anaerobic on the same time. So both O and F positive. But on the other hand, we have Pseudomonas, which is strict aerobic, which means that it will grow only in the presence of oxygen. So it's oxidation positive, but fermentation negative, which means that there will be no utilization of, if you don't apply oxygen, which means that the tube without oxygen, it cannot utilize the fermentation of sugar process to produce energy, and that's why it will remain green. So it's O positive, F negative. Now, this is very important classification. Again, it's another type of fermentation process because it's a different sugar. So in the previous test, we spoke about glucose. Here we are talking about clad agar, which contains lactose. So a lactose fermentation is very important process in which the bacteria can be grouped into two main groups. Lactose fermenters that will turn the color of the clad from blue into yellow. So lactose fermenters are mainly E. coli and Klebsiella pneumoniae. So both bacteria can utilize lactose in the medium and then it converts to yellow. On the other hand, we have other groups of Enterobacteriaceae that are non-lactose fermenters, and it's actually the longer list that contains all the other Enterobacteriaceae, including Salmonella, Shigella, Proteus, and Pseudomonas aeruginosa. We will learn more about a little bit more details in a minute. Now let us learn into individual bacteria, the specific characteristics for each. For E. coli, we call it, the full term of E. coli is Escherichia coli. Some of them call it Escherichia coli, but doesn't matter. We call it E. coli for short. So what is the main source of E. coli? It's actually E. coli is found in our normal gut flora, and it's also found in not only in humans but also in animals, and that's what makes the transmission from humans to animals or from animals to humans also happening like a round cycle all the time. And the bacteria, if it's in the feces, is actually can be viable or alive for a few days, and if the water is contaminated, then the person can get sick. So you can see here very clearly from the picture how we can get easily infected by E. coli from oral fecal route. So that's why if you eat undercooked vegetables, undercooked meat, or if the water is not treated or properly filtered and it's contaminated, or unpasteurized dairy like milk, then we get sick. So how does E. coli becomes virulent, and what are the most important virulent factors? So, what are the most important diseases caused by E. coli? So, you'll see me that throughout this lecture, whatever we are going to talk about the diseases caused by different bacteria, we call it clinical importance. For E. coli, the most important or significant disease that is caused by it is called gastroenteritis. And from the term itself, you can conclude that gastro means stomach, entero means intestines. So it's the diarrheal illness that happens in the gastrointestinal tract. For E. coli, we divide them into two different types of diseases. The first one is enterotoxigenic, which means that it's a group of bacteria or serotypes of E. coli that is capable of causing disease in the small intestines, and this will result in watery diarrhea. So the diarrhea or the loose motion will be like water. The second type is enterohemorrhagic, that happens in the large intestines and usually involves the bloody diarrhea, and you are going to learn them into details later on. Now, to continue on the most important, clinical important gastroenteritis diseases, The enterotoxigenic E. coli is also known as ETEC for short, is since it's actually happening in the gastrointestinal tract, it's transmitted by water, and also it's known as traveler's diarrhea because it's very common when you travel to endemic areas like developing countries. And this happens because the bacteria has different virulence factors, especially the heat labile toxin and the heat stable toxin. So the first one, or the heat stable toxin, is increases the cyclic GMP. The second one is the heat labile toxin, which is similar to another disease called cholera, and it increases the cyclic AMP. And this is, we're not going to go through the details, but these are the most important virulence factors for these type of E. coli. The second one, which is the enterohemorrhagic E. coli, is known as EHEC. And to actually, uh, be able to remember this, it's actually transmitted to a human when you eat undercooked meat or hamburgers. And to remember this, you can also see the common here is H in EHEC and H in hamburger. Not only gastroenteritis, the most common disease also that E. coli can cause is urinary tract infections that we call them UTI. So why does it happen? Because actually the bacteria has pili as virulence factors as well. And these pili will make the bacteria able to adhere or to stick to the uroepithelial cells. And then when it sticks, it will be able to also move by the flagella from the urethra to the bladder and spreads the infection. It's more common in females because, of the shorter urethra and also the proximity of the urethra to the colon. So that's why it makes it very common. Another disease is meningitis or neonatal meningitis, which means meningitis in newborns. And this is mainly because of the presence of the capsule. And this capsule makes it pathogenic. And this, what we have learned also earlier, is called K antigen. Now let us move to the second group of bacteria. Klebsiella pneumoniae is a very common pathogen also, and we see it also on a daily basis, unfortunately. It's a facultative anaerobic bacteria, but also it grows back better in the presence of oxygen, so it likes oxygen. So that's why I want you to remember this, and it makes it very easy for you to remember. Any place where we have abundance of oxygen, Klebsiella will grow. So you can find this actually as part of the normal gut flora and also in the oropharynx. So we have it in the oropharynx and we have it in the gut flora in the intestines. Not only that, but actually it causes a disease from the bacteria name itself, which is called pneumonia. So the disease is also pneumonia, which means infection of the alveoli and the collection or accumulation of fluid inside the alveoli. And that's why we call it also hospital-acquired pneumonia, because it's mainly happening inside patients in the hospital, but it can also be in the community. And because it likes oxygen, it grows also in patients on ventilators. So patients who are receiving artificial oxygen, because there is continuous flow of oxygen and unlimited flow of oxygen, it will make it grow and also colonize these ventilators. A common disease also for Klebsiella is UTI or urinary tract infection. Actually, it's the third most common cause of UTI, and also septicemia, which means sepsis in the blood or the bacteria presence in the blood. Okay, so Klebsiella, as part of the big family Enterobacteriaceae, is actually gram-negative bacilli. And these are very short and plump bacilli, as you can see in this picture here. As you can also notice in this picture that they are surrounded by a halo, and this is actually a capsule. So they are encapsulated or capsulated. And a very important also for us during the lab is that it's non-motile. But it's actually, this capsule make it very mucoid. So you can see here from the picture as well that it's very sticky to the plate. It grows and it also occupies the whole plate because of the stickiness or this mucus layer because of the capsule. Now let us move to the other type of bacteria, which is Proteus. So how this E. coli becomes pathogenic? This slide here, we can see that we have learned also before that we have the gram-negatives have the endotoxins, but for E. coli and these Enterobacteriaceae, there are more. And it's very important to learn about these structures because these give the bacteria the antigenicity, the virulence, and on top of that, the ability for us to differentiate the bacteria and group them into different serotypes or genetically variables. And the main groups for the E. coli is actually capsule, which is the K antigen. In case of Salmonella, it's called VI antigen, somatic or the O antigen, and the flagellar or H antigen. So the capsule is very important virulence factor because it protects against phagocytosis, and also the somatic one is the endotoxin or the LPS part of the endotoxin. And these are very important because when we classify E. coli, we actually give it the different sero group numbers, and you're going to learn them into details when you learn the systems by next semester and also next year. So what is Proteus? Again, it's part of the big family Enterobacteriaceae. These are, again, gram-negative bacilli, but they are pleomorphic, which means that they can be sometimes present in the form of short, plump colonies. Sometimes they are more longer, sorry, not colonies, I meant they are short rods. Sometimes they are longer rods. Sometimes they are similar to cocci, so they are cocobacilli. We call them pleomorphic. And there is very important distinct characteristics for Proteus is that it has the ability to move, or it's a motile bacteria, and because of that it swarms the type of motility known as swarming. And this can be seen on a blood agar because the bacteria can move easily. And you can see it here, like it looks like a flower flourishing on a blood agar. This motility actually again makes Proteus able to descend over the urinary tract, causing infection, and it's one of the most common causes of urinary tract infection. So far we have learned E. coli, Klebsiella, and Proteus as three main causes of urinary tract infections. And a very important characteristics of Proteus is through biochemical tests. It's a urease positive bacteria, and it's H2S positive as well. So basically urease means that the bacteria has the enzyme urease, which converts urea into ammonia, and then it will turn this alkalinity or increasing alkaline medium changes the pH of the medium, and this will change the color of the medium. And H2S is actually hydrogen sulfide that is actually formed when the bacteria also can convert or it has a specific enzymes for this reaction to happen. And also the most important characteristic of Proteus, it is positive for phenyl pyruvic acid test. As you can see from this picture here, it among others, it's one of the most important tests because it distinguishes this tribe Protea from the other Enterobacteriaceae. So if you are suspicious because other bacteria are urease positive or H2S positive, PPA or phenyl pyruvic acid can actually give us the correct conclusion that this is Protea species. Okay, now let us move on to the other types of bacteria. So after finishing with the most common causes of UTI or urinary tract infections, now let us move to more other type of bacteria that can cause diseases like gastroenteritis or disturbances in the GI tract. The first group of bacteria here I want you to talk about, which is also again part of the Enterobacteriaceae, is called Shigella. Can you see here the picture on the right side, hand side? The Shigella is saying, to the toilet you will run when I have my fun, which means that this bacteria actually is very mean. It can cause very bad diarrhea. And you can see also in the other picture on the top here, the bacteria is very sleepy, and in a moment you learn why. So Shigella has very important feature. It's a lazy bacteria, or we can call it biochemical inert bacteria because it's a gram-negative, it's non-motile, it's a non-lactose fermenter, it's oxidase negative, and it does not produce a hydrogen sulfide on on a media like the triple sugar test, so it will not form a black color. It will only give you a negative for almost all the tests. That's why we call it lazy bacteria or very biochemically inert. And this is very important for you because you also will be able to remember the difference between it and the other groups in the family. Okay, so we learned that Shigella is a gram-negative bacilli. They are very inert, non-motile, and also they are biochemically inert. There are four important species in this group. So we have Shigella dysenteriae, we have Shigella flexionare, we have Shigella boydii, and Shigella sonii. So all of the above three are non-lactose fermenters, except the last one, which can be a late lactose fermenter, which means that it can ferment lactose, but only later on, which is after some time of growing in a media. That's why we call this bacteria also biochemically inert. And again, it's a catalase negative. The most important disease caused by Shigella is called shigellosis. Shigellosis basically means dysentery or bloody diarrhea. And also we are going to learn more about these diseases later, but we are going to mention a few of these characteristics shortly. A very important virulent factor for Shigella is called Shiga toxin from the name itself, and this Shiga toxin is actually have the ability to inhibit protein synthesis. And now let us move to the media in which we grow Shigella. So the media, the most important media that we use is actually very similar to the ones that we use for the next group we are going to talk about, which is Salmonella. Selenite F is an enrichment media in which there are a lot of inhibitory factors inside the media that will allow the inhibition of all the other groups of the commensals in the gut, but only allow the growth of Shigella and Salmonella and very specific species. McConkey agar is again a mildly selective agar, which means that it only allows the growth of or selects for the growth of specific groups of bacteria. And for Shigella, it produces those colorless colonies. They are like yellowish, a little bit yellowish colonies, while because it does not ferment lactose, because if it was a lactose fermenter, it could have changed the color into pink. And the third one is the moderately selective media, which is XLD. So XLD is used also for Salmonella, and we differentiate between them by the color of the colony. For Shigella, it produces pink colonies. And for Salmonella, it will give you a different color. Okay. Now let us move on to Salmonella. And when we talk about Salmonella, we actually have different species. So we have Salmonella typhi, Salmonella paratyphi, Salmonella typhimurium, and Salmonella enterotoridis. And we are not going to go through so many details. But I want you to remember one very important story about Salmonella. This actually a bacteria that became very important, and it was named after the famous story of Typhoid Mary, and that's where the disease typhoid was also came from. Actually, this lady was used to work as a cook, and she used to work in houses. She cooks meals for people, and it was noticed that this lady, every time she cooks for people, they fall sick, and most of them die. but she was completely healthy. And it was thought that this lady actually kills people whom she was working with. But then they found out that actually this lady was not killing her people that she was used to work with them. Uh, she actually was, I think, present for some time. You can read her story in the internet. But then it was discovered by some doctors that this lady actually carries in her guts type of bacteria. So you can see here that some people are chronic carriers of some bacteria. She did not have any disease, but she was spreading the disease to the people through the contaminated hands. So it was bacteria that is stored in her body in the gut and the gallbladder, then excreted by the feces, and then through the contaminated, unwashed hands she used to cook for the people. The people got sick. Very sad story, but actually we learned a lot from this story. So Salmonella is a group of bacteria that cause diseases of the gut. So we divide them into two main groups: the enteric fever group, or the salmonellosis, and it's basically the first two bacteria, Salmonella typhi and paratyphi, and the food poisoning group, or gastroenteritis, which includes the other two. So salmonellosis is caused mainly by Salmonella typhi, but it's a much more milder disease when it's caused by Salmonella paratyphi, and it's basically means that it's similar disease, but it's much less in severity. So what is typhoidal salmonellosis means? It's typhoid fever or enteric fever. So the patient will have fever, then constipation. Constipation will be followed by diarrhea. And some of the patients will have rose spot rashes on the skin, not all of them. So this is what I want you to remember as far as this lecture is concerned, but you can find more details if you need to. The other salmonellosis can be caused by non-typhoidal strains or species. We call them Salmonella enteritis and Salmonella typhimurium. And these two bacteria invade the ileocecal region of the gastrointestinal tract and cause inflammation, increase in the cyclic AMP, and then this results in a type of diarrhea, which is watery diarrhea. And that's why actually this is very mild. Gastroenteritis is much more easy to deal with compared to the typhoidal salmonellosis. Now we discuss the clinical characteristics of Salmonella typhi. So these are gram-negative rods. They are motile, so they are different from Shigella. They are again non-lactose fermenters, similar to Shigella. They are oxidase negative because they are Enterobacteriaceae group, and they can produce H2S, which is hydrogen sulfide. So it will give you a very important characteristic that we will mention in the next slide. So since it's a non-lactose fermenter, on the clad agar, what you will see is colorless colonies. Okay, so for Salmonella, these are the gram-negative bacteria that we see under the microscope. And I also want you to have a look at this video here to see the motility of the bacteria. You might not be able to see the video here, but I'm going to attach it into Moodle. You can have a look at the different motility, and how we can demonstrate this in the lab is actually we can grow them under in the agar, and then we put spots under the microscope, and we can see actually this motility very well. There's another test that can actually differentiate the different Enterobacteriaceae, which is the indole test. Indole test basically is that a media that contains a specific type of chemicals or growth media, if the bacteria has the indole, it will be able to form a pink or magenta type color ring on top. If it's negative, it will not change. So this is actually an indole negative bacteria. I'll show you a slide at the end on how we can summarize this result for all the bacteria. Salmonella can grow in enrichment broth. We call it Salinite F, same as for the Shigella. So it's the same broth. But also we can use another broth called tetrathionate, and it's a broth that can convert from the blue color into green color. That indicates that there is growth of Salmonella species. Other media we can use for Salmonella, it's very easy to remember because they are similar to Shigella. So for McConkey agar, again, it will be colorless, give you colorless colonies. So that's why we cannot distinguish it at this stage from Shigella. So we need another media, which is XLD. In Shigella, we see that Shigella will produce pink colonies, but Salmonella will give you black colonies. Why? Because it's H2 S positive. So it means that it can convert it can produce hydrogen sulfide that will give you a black color on the media. So you can see that these colonies immediately they are salmonella. On the triple sugar agar is actually what you can see again. It's a black color because of the H2S positive test. A third media, which is actually very highly selective, is the Wilson and Blair. And it's very selective because it actually converts the black colonies. It's very clear. Other bacteria will not give you the same result. Now, in this picture here, I just wanted you to see all the different tests. How does the chemical reaction happens? And I don't expect you to know the details for each test, but I want you only to understand it. But I want you to remember the table so you can differentiate between the positivity of each test, like the citrate, the urease, and the indole. What we use to do what we used to do previously is that we to do all of these tests together, and we wait for the results, and then we interpret them collectively. So we don't use one test or the other. Actually, we use all of them, and then we interpret each test so that we can compare. So each group of bacteria will give you a certain positive or negative result, and then we can conclude which is the most likely bacteria. And then after concluding from this biochemical test, we can proceed to the more specific tests in the other detection methods that we have in the lab. Now let us move to another important group of bacteria. We call them Pseudomonas aeruginosa. This picture is a very nice picture because it always reminds me that we have it tells us a lot about this bacteria. So this bacteria is saying, splish splash, I'm right here in your bath. So you can conclude from this that this is a water-loving bacteria that is found in the environment, mainly in ponds, in the soil, on plants, or sometimes in fresh fruits and vegetables. So for us as human beings, it's very important as well because it can cause a lot of diseases, especially when there are moisture. So that's why it's present in the ear if you swim on swimming pools, and that's why we call it swimmer's ears, or the inflammation or infection in the external ear called otitis externa. And we have opportunistic pathogens also in the hospital because this bacteria can grow easily in patients with low immunity or who are immunocompromised. And that's why we call them nosocomial, because they are acquired in the hospital. Mainly, it can cause sepsis. It's a very common cause also of urinary tract infection and also wound infection in the burn unit. The most important characteristic of Pseudomonas aeruginosa is that it's a non-fastidious. It can grow at any temperature, especially 42 temperature. It's obligate aerobe, so it loves oxygen. And you can remember aeruginosa, A stands for air. And it's a non-lactose fermenter, so it does not produce any change in color on the clad. But it's very important for you to remember that it's oxidase positive. So this is the main difference between Pseudomonas and the other group of Enterobacteriaceae, which are all oxidase negative. A very important distinct characteristics that we use in the lab is the smell or the scent of the agar plate. This bacteria smells like grapes, and on the nutrient agar, which is yellow, it easily can be converted into blue or green dye because this bacteria produces a pigment called bioverdine or biocyanine, and also there's another additional uh, dyes here like biomelanin and biorubrin. So this bacteria is very rich in pigments. And this here shows you, the picture shows you how does this show you, or this pigment shows very beautifully in the nutrient agar. Now let us move to another group of bacteria called Vibrio species. So Vibrio is a curved or a comma shape. And this comma shape is actually gives a distinct appearance to the bacteria. It's a gram-negative rod, and it's very characteristic of a very severe disease called cholera. And this bacteria produces an enterotoxin called cholera toxin. And again, it's a gastrointestinal disease, so it's transmitted by the feces. So if you drink contaminated water or food, you can easily get sick with Vibrio, especially in endemic areas like India or poor countries. And we have two very important species in this group for you to remember. The Vibrio cholerae, which causes the severe diarrhea, and the Vibrio parahemolyticus, which causes milder form of gastroenteritis. So this picture here shows you nicely how this curved bacteria looks like under the microscope, and some of them actually you can see the flagellum at one of the ends. A very important characteristics of Vibrio cholera is that it is once it infects people, it causes what we call rice water stool. So the stool looks like rice water, so it's like very light and very loose, and it can cause very severe form of disease resulting into dehydration and electrolyte imbalance, and this can lead to very fatal condition, cardiac and renal failure, and a very high mortality rate, which means that some patients might die because of this infection if not treated immediately. The mortality rate is up to 40%. You can see here also on this agar on the right hand side, it's a very selective agar for Vibrio. We call it CCBS, and it converts, if it's cholera, Vibrio cholera, it converts the colonies into yellow. If it's parahemolyticus, it will give you the green colonies. Okay, so as I mentioned, the curved bacteria. So we talked about uh, the Vibrio before. This is Campylobacter, which is another group of a twisted bacteria. It's a gram-negative curved rods. They are again fastidious bacteria because they have specific requirements, and they are the same group of bacteria that can also cause dysentery. And it's a type of zoonotic infection because it happens in animals or can be acquired through animals, especially contaminated chickens and goats and cattle. And the most important characteristics of these or more common important findings of Campylobacter is that we find it in developed countries and less common here. And virulence factors that this bacteria has is the flagella, adhesins, and also endotoxins. They become very invasive, especially the enterotoxins. They are very invasive and can penetrate the cells of the mucosal lining of the gastrointestinal tract. Now, I think this is very important slide for you to remember is that bacilli or gram-negative bacteria are a very large group of bacteria. If we want to differentiate between them, we have to grow them on a lactose-containing medium like lead. So if it's a lactose fermenter, then it's E. coli. If it's a non-lactose fermenter, then we have to perform oxidase test. It's a very simple test. So if it's a positive, then it's Pseudomonas. If it's negative, then it's we have to ask ourselves, is it producing H2S on the triple sugar agar or the XLD agar? Then it's Salmonella. If it's not producing H2S, then it's Shigella. Another important characteristics that we look at is the motility, because as we said earlier, the some bacteria are motile and some are not. And the mnemonics that we use for this is SHIPES. A very important uh, laboratory diagnostic features is that they need chocolate agar plus antibiotics to grow, or Skiro's blood agar. But we have in our lab the Campi blood agar, which is like a very black agar, and it is grown under incubation conditions that are called microaerophilic, which means basically a specific requirement of oxygen tension, which is 5% oxygen, 10% CO2, and 85% nitrogen. And it grows also at 42 degrees Celsius. The most important biochemical identification test for this bacteria is oxidase test, and it's oxidase positive. So again, this feature differentiates between it and the other Enterobacteriaceae, and also catalase, which is catalase positive in that case. A very important virulent factor for Haemophilus influenzae is that it can produce IgA protease enzyme. At this degradation enzyme can degrade the IgA, which is produced in the mucosal surfaces as part of the immune system. And a very important another factor is the polysaccharide capsule. And this is also important for the serotyping of the bacteria. It gave us 6 types: A, B, C, D, E, and F. So these are very important, but they are very also virulent and invasive than non-encapsulated. And the most invasive one is the B type. The most important disease caused by Haemophilus influenzae is serotype B causes hip epiglottitis, which is infection of the epiglottis, meningitis, and pneumonia. A quick note on Haemophilus influenzae, because it's a very important gram-negative cocoa bacilli. And we call them cocoa bacilli because they have both appearance of either cocci or bacilli or something in between. That's why they are pleomorphic or having 2 different shapes. And from the name itself, Haemophilus means they are blood-loving bacteria, and they require blood because they need it to grow. So they need 2 factors to grow: the hematine, which is called factor X, and the NAD, which is factor V. If these are present, then the bacteria is happily going to grow. The most important distinguishing characteristics of these bacteria is that they are aerobic, so they require oxygen, and they are gram-negative, and they are catalase oxidase positive. So they are unlike the Enterobacteriaceae, and it's similar also to Pseudomonas. It's oxidase positive. And as we mentioned earlier, blood agar does not have the NAD factor. So blood agar needs to be heated to produce chocolate agar so that the bacteria can grow easily, because the lysed blood will contain NAD or factor V. So this bacteria is fastidious, so it grows in CO2 enriched incubators. So this is a very specific requirement. Otherwise, the bacteria will not grow into happy colonies. And there is a very important phenomena that we see with the Haemophilus influenzae, which is called satellitism, which means that the bacteria can produce only in blood agar. They can grow only if you provide them with the bacteria that can lyse, or the bacteria can do hemolysis like Staph aureus. So if you put a line of Staph aureus, Haemophilus influenza can grow around it because now blood is lysed. As we approach the end of this lecture, I would like to thank you all for listening and also for your patience. It's quite dense lecture, and although we finished the lecture, but I feel like we have not covered it very adequately, just touched on it, and I know that it's not because because of the time limit and also because you're going to learn these into more details in the future. I have put here very important resources for you because I think these two YouTube channels are very important for you, not only for microbiology, but you can find also very useful resources for medical students and for the biomedical scientists also. So thank you very much for listening. And also you can find that a lot of the pictures I've taken today in today's lecture are from the ATP YouTube channel. So please, if you have any questions, we can discuss further. You can send me any emails. Thank you very much.